Oh, share my desktop. Yeah. So, Hemant, can you confirm? Okay, you can see my screen?
Yeah.
All good? All good? Yep, yep. All right, I'll start. And before I do that, all right, yep. Hello, everyone. And I would like to thank you, Professor Hemant, for inviting me for this conference. And for me, this is a very special conference. Two of my mentors are part of this conference, and I consider that as a privilege that I'll be speaking at the same conference where they are. One of them is Professor Ivan Scutino, whom I know since my B.Pharm days, and eventually I did my PhD. And the second one is Professor Bhartam, from which I learned a lot of quantum chemistry while my time at NIPER. So what I'm going to talk today is understanding how a drug binds to its target and atomic level. Now this presentation is more going to be like a story of some of the work I have done and we have done as a team over the last 10 years, my time in Australia. So the overall theme of the work is we want to understand how a drug binds to its target. For example, it's shown here on the screen. Sorry, I'm just trying to get the, yeah, okay. So here, we get a lot of experimental data for protein ligand complexes, mostly coming from X crystallography, NMR, electron microscopy, mass spec, and glycan arrays. Since we cannot see those structures by experiments, they are combined with the computational models and force field parameters to generate the final structures, and which we use for, of course, drug design and design of therapeutics. So the overall theme of the work has been to using this computational approach for three purposes. First is to interpret and reconcile the experimental data. Second is to complement and guide the experiments. And third is, if possible, we can replace some of the experiments. So most of the technology I have used for the work I'm going to present is a standard molecular dynamics simulation, just showing a small movie here, looking at the dynamics of atoms and interaction with the rest of the system and how we can evaluate and get some numerical data to make sense of what is happening at an atomic level. The second critical part of the simulation is the force field parameters, which we use to describe the whole system, which is basically composed of two components, bonded interactions and non-bonded. And bonded terms come from the bond stretching, angle banding, rotation around the bonds, and the keeping the groups planar. And the non-bonded are comprising of two major components, electrostatics and van der Waals. And most of the work which I'm going to present today involves another technique, which is the free energy calculations. Now there are two ways to calculate the free energies. One is called thermodynamic integration method, where you calculate the work, to be honest, reversible work required for a given process. And the second method is the thermodynamic perturbation. In this method, you calculate the probability of finding a given system in a given state. For example, in practical sense, what you do is look at the two ligands, which are A and B. And you want to calculate, for example, delta G2 and delta G3. Now, delta G2 and delta G3 in slide represents the experimental binding free energy when a molecule transfers from water to the protein binding cavity, which is done by experiments. Now, this can be done with the simulation, but it's very computationally intensive. So what you do in your calculation is calculate the relative free energy between the two states performing alchemic mutation. In this example, you calculate delta G1 and delta G4. In delta G4, you're doing the transformation of molecule A to B in water, And in delta G1, you are doing the same transformation in protein and you get the relative binding free energy. And again, these calculations are not just simple or straightforward calculations. What you have to do is, because this process has to be reversible, when you make a transition from molecule A to B, it has to be done in a large number of steps which are reversible at each stage using something called coupling parameter approach. And in a crude sense, I just show the plot here, which shows that just to calculate one free energy number between two compounds, you may need to run more than 10, 15 individual calculations, obtain the what we call the free energy derivative at individual lambda points, going from state 0, which is ligand A, to state 1, which is ligand B. Integrate that curve and you get the free energy number. Now, although this is computationally intensive, but this is one of the most accurate method. Of course, it's based in statistical thermodynamics or statistical mechanics, and had been of a lot of interest in terms of drug design and discovery. But there are two major challenges in this method. First, this method takes a lot of time in terms of computing time to set up and run. So what we did at the time, that we wanted to have a high throughput method where we can get this number not by approximate method, but more by rigorous free energy calculation methods, but in a short span of time. So we said, all right, we will develop some method and optimize some of the free energy calculation methods so that we can have a rapid throughput. So first thing we need to do is we need a reference data set where we know the exact answer from the calculation, and then we can try alternate methods and try to reproduce the same answer. The first data set we tried was glycogen phosphorylase, now which is an interesting target for diabetes, and there are more than 50 high resolution structures available in the public database, for example, Protein Data Bank, and we have a lot of biological and inhibition data available. And now this represents a nice data set because we have a lot of structure, a lot of binding data, and we can develop and validate any of our methods. And all right, this is a good starting point. So one thing we noticed when looking at those 50 structures, now we noticed there were two structures of the same ligand with the same protein here reported on not 2 years apart by the same research group. And it has a difference. We thought it was a minor difference. And if you look at the picture here, which is a red circle, now red represents oxygen and the blue is a nitrogen. So it's an amide group in the ligand. And in the second structure, the amide group is flipped, and simultaneously the asparagine side chain on the right hand side is also flipped in the protein structure. Now, these are just alternate representation of amide molecule. But again, it looks simple that, okay, this looks simple, what's the issue here? What happens that we can't resolve the structure from electron density at a given resolution at 2 or 2.35. And again, those two interactions are complementary because when I flip the amide on my ligand, the amide group on the protein is also flipped. So there are complementary hydrogen bonds in both the structures. But one of them has to be what we call thermodynamically more stable. So what we decided to do, let's calculate the binding free energy between these two states and see which one is the most stable structure and if there are any implications. So first thing we did was, because this molecule is inhibitor is a glucose analog, and it has an amide group attached to the glucose molecule. So we just look at the rotation of that amide group using quantum mechanics method in isolation. And what we see is this plot here. So what we find is for that molecule, which is shown here on the right under 1GG8, we look at the rotation of the amide bond attached to the ring. So we find three minima here. One is the minima when the torsion angle is 0 degrees, which we call QM minimum 1. Then there's another minima at 300 degrees, which is QM minimum 2. And there's another minima at QM3 at 240 degrees. Then what we did, we had the similar compounds from those 50 crystal structures and tried to see where those structures fit in actually. So when we took the structure out of the 1GG at crystal structure, it was actually sitting at the quite higher up in the energy on the potential energy diagram here. Then we took the structure and took to the one of the validation server available at the time, which is not available anymore, Welding URL and the hiccup. And now that structure actually gives the structure sitting at at the highest saddle point in the potential energy surface. But one thing we realized that if we take these structures and just swap oxygen with the nitrogen, actually we do find a minima. So if I take the crystal structure 1GG8 and I swap oxygen with nitrogen, actually it is the QM minimum 2. And if I take the structure from the validation server and swap oxygen nitrogen, actually it is the QM minimum 1, which is the global minimum. And again, this is just to highlight that oxygen and nitrogen are not easy to distinguish in electron density when you solve a structure using X-ray crystallography. So it's easy to make this mistake of an amide, and it's just a single amide group. But what happens that if you make one amide group in a particular way, the whole hydrogen bond network within your protein structure or with your ligand and protein interaction totally changes, and it can have different consequences. Now we realize, all right, making this swap can have a significant difference in the energy when they are free or in isolation. Now our interest was that whether this would make any difference if that difference was, or that oxygen and nitrogen atoms were placed in the protein binding cavity. So next we did was we took up this minima and some of the structures we had, put it in the protein binding pocket, do systematic free energy calculation using thermodynamic integration using multiple lambda points just to get one number and see if it makes sense. So what we find here that we build actually few closed thermodynamic cycles. Now the reason for that is that when a closed thermodynamic cycle, for example, you go from state 1 to 2 to 3 and come back to 1, that overall change in the free energy would be 0. And that is one of the ways we are able to test whether free energy numbers we have obtained are converged or not. So here we build actually two thermodynamic cycles between multiple states as shown on the picture. So what happens is that the conformation which was assigned in the crystal structure 1GG8 shown in the top of the figure here, that was the highest energy structure with the roughly 22.6 kilojoules per mole with the binding free energy difference. And actually, if we take the crystal structure and I swap the oxygen and nitrogen, that is the lowest free energy binding state. So what we found here that just swapping oxygen and nitrogen in that molecule could have a difference in the free energy up to 20 kilojoules. And that was significant. So we said, all right, let's look at because there are more than 50 crystal structures with a similar compound for this molecule, and go back and see where do they sit on the potential energy surface. So here I just shown the 5 structures of more than 5 here, and just place them on the plot here. And to our surprise, we could not find a single structure of the ligand which was showing the one which is shown on the right circle. That was the global thermodynamically free energy minimum in the binding pocket. So none of the structures we had represented that conformation. And then we looked deep into how these things are done. So we noticed that, of course, all structures came from the X-ray crystallography. And of course, it's an indispensable tool in the structural biology. And one of the key things we realized that most of the structures contain, of course, biomolecule like protein, and some of them are nucleic acids and sugars and lipids. So we have highly refined parameters for those biomolecules for their refinement. So the structures of proteins and most of the nucleic acids are solved near atomic detail. And we have a lot of validation tools for protein structure to check its structure, quality, and geometry, like ProCheck and mole probability. But when you have a small, non-covalently bound organic molecule in the binding pocket, those structures could be very less certain. And for example, like protein data bank is updated every Tuesday of the week. And as of this week, now we have about 170,000 structures. Out of this, 90% of the structures come from X-ray crystallography. And out of that, roughly 35,000 structures contain at least one of the small organic ligand molecule. And what is the limitation here? We try to work out, okay, why we are getting discrepancies in the structure of the ligand. For example, if I look at the function of electron density and how the structures are solved, for example, if the extra structure is solved at very high resolution, the lower is the number, higher is the resolution here. So for example, here the 0.66 angstrom. Now the shape of the electron density is so clear, we know that is a tryptophan side chain. And we can place the indole molecule very nicely. But as the resolution drops, the density becomes kind of much smeared and it's not easy to place the group clearly. But that is not an issue for the proteins because what happens, of course, the amino acids are connected to each other. When you solve any structure, you have a prior knowledge of the amino acid sequence. Once you are able to solve the part of the structure, rest of the structure is much easier and pretty straightforward. But when you have a small ligand molecule, which is non-covalently bound in the binding pocket, it gets very tricky. For example, here, one of the examples I've shown is phosphorylic acid derivative. At a high resolution, which is 0.97 in this case, you can clearly see the electron density. You can build your structural model just based on density. But as the resolution drops, then the structure you build is governed by the geometric restraints. And when I say geometric restraints, it's more about how you model those compounds, how you build those structures, and what kind of parameters you use for the refinement. As the resolution drops, it becomes very difficult to assign what is the orientation of my molecule, what is the conformation of molecule. And of course, we cannot see protons at that resolution in the extra structure. So we don't know what is the tautomeric or protomeric state. And all these are very critical for the drug design. For example, in this case, we know at the higher resolution structure how the ligand should be binding here. But if in the absence of that structure, if we had structure only at say 2.4 or 3 angstrom, and this is the way ligand was placed based on the bias from the structure building and the structure refinement program, then you are more likely to fail on your drug design effort using that structure. So the big question here is that, of course, once the structure is solved, how do you validate? So what we have done over a time, we have developed multiple protocols that once you have the right force field parameters, we can perform molecular dynamic simulations and free energy calculations and then work out what is the most probable binding mode of the ligand molecule. Now just I'm showing you one example from the literature, which was a very, very interesting one. Now this was reported by a research group which were working on the PPAR receptor, quite a hot topic 10 years ago. And many molecules were targeted for diabetes and obesity treatment. Now they had solved a structure which was called 2GWX in the protein data bank. And they claimed that this is the upper form of the structure, means there's only protein, no ligand. But this upper form has been crystallized in the activated state as if the ligand is bound. And the same research group, they redefined the same structure after 2 years based on certain information they had. They realized that the chain of water molecules they see in the binding region of one of the binding regions actually is not water. Actually, there was a small fatty acid trapped into the binding pocket, and that was the reason this was in the activated state. And again, the reason I'm showing this example is that, of course, experimental methods are all good, but once you solve the structure using the X-ray and NMR, once you get the data and build this density or the noise, that's where the experiments stop. And what you do after is all computational and structural modeling. And what structure you are going to get will be governed by the quality of parameters and the methods you use. So one of the big factor which came in was that not only the geometric parameter of the small molecules, but also the interaction parameters, what we call the description of the molecule or the force field of the small ligands is very critical. If we don't have the right parameters, it's less likely we are going to get the right structure. So when we look at in literature, most of the force field parameters will be used in the structure refinement, molecular simulations, and drug design. Now we have so many biomolecular force fields which have been developed over last many years, and systematic improvement for proteins, nucleic acids, and lipids, and to an extent sugars. Now what we are talking about here is we want a description of the force field for small organic or drug-like molecules, not only by itself, but it has to be compatible with the given force field. So what we have done over last few years, that we have taken the Gromos force field, which has been validated against thermodynamic properties of small molecules. And what I did was, at that time, I developed a semi-automated protocol that we take a small molecule, we do a series of quantum mechanics calculations, then we integrate the parameters from the biomolecular force field and generate the description of that small molecule. And then we named it ATB, Automatic Topology Builder and Repository. Now this is just showing the web interface of the ATB, and on the right hand side is showing the schematic or the workflow, how the molecule is processed through various levels of theory and what parameters are obtained. And this actually was started in sometime in 2011. And at that time, we only had about 500 molecules. Now, over a time, with a lot of development and validation work, currently we have about over 13,000 users worldwide, and we cross about 450,000 molecules in the repository. Now, these are the protocols which work in the back end of this web server, and you submit any molecule. If molecule is already in the database, it will show up as a result, or else it will say it's going to process the molecule in next couple of days and produce the results. And the output parameters could be used for, of course, in docking, drug design, and the structure refinement. So recently what we did a couple of years ago, that we need to do is, of course, these are the just parameters. How do we validate? So one of the key things we had an interest was we wanted to reproduce thermodynamic property, for example, the binding free energy. And one of the key aspects for the calculation of binding free energy is that we should be able to reproduce the hydration free energy or the solvation free energy of small molecules. So we took a data set of simple single substituted molecule of about 357 molecules and calculate the hydration free energy for a different chemical class and compare with the experiment. So this is not the final, this is not the initial plot, this is the final plot. So initial plot was not that good. We had certain chemical groups which were scattered all over. So what we did was we systematically pick up those functional groups, optimize those force field parameters, what we call parameterize them, and then rerun and then reproduce the hydration free energy. Now the reason I'm highlighting more on the hydration free energy is when we look at the right, when we do the free energy calculations in computational work, what you are doing is calculating the binding free energy between the two molecules in proteins and two molecules in water. Now here we know the proteins and the small molecule force fields have been developed with a similar strategy and they are compatible. So the accuracy of getting that number is much higher. But if we make any small mistake in the hydration free energy of individual molecule, we are more likely to get delta G3 incorrect, and then overall binding free energy incorrect. So to have this success in the free energy of binding between two molecules, we have to make sure we are able to reproduce hydration free energy of these molecules. So now once we have developed protocols and web servers that we can produce force field parameters or description of small molecules, then we look at the next data set, because we still wanted to have a data set where we can reproduce binding free energy data, and then we can use that as to develop and validate high throughput methods. So we went back to our initial aim of developing the better throughput free energy calculation methods. Now this time we took a data set of a protein called PNMT, phenylethanolamine and methyltransferase. Now, the reason for choosing this data set was that at the time we had an experimental collaborator on campus who had solved in their group more than 42 structures using crystallography and in complex with small drug-like molecules. And they had access to a lot of biological data. So what we did the student we had that we took all the structures from PNMT and drug complex and run what we call the molecular dynamic simulation of the complexes using the parameters from the ATP and see how those structures behave. And almost all the structures are pretty much stable in the simulation, which indicated, of course, the structures were solved at place in appropriate manner in most of the structures. But there were a few structures which were unstable in our simulation. And then we said, let's go back and investigate what's going on with those structures. So the whole purpose here was that in case what we wanted to do, we wanted to identify what is the preferred orientation, conformation, even the stereoisomer. Now this is critical in the cases because many structures are solved using the racemic mixture of the ligand, and we want to, the density won't tell us whether which isomer has been crystallized or maybe both of them are, crystallized. And what is the protomeric and tautomeric state? And to investigate this, we looked at this one of the systems from the PNMT, and here just I took up this example just to demonstrate how critical this will be. So what happens here? S-noradrenochrome on the left-hand side, actually it's an inhibitor of that enzyme, and on the right-hand side is the R-noradrenaline, which is a substrate for that enzyme. And the two crystal structures are solved. Now there's a drastic difference between the two structures. What happens on the right-hand side is a substrate. Now substrate has been crystallized using the R stereoisomer and has a particular binding mode as shown here. The structure on the left-hand side is the inhibitor. Now there are two differences. Now the inhibitor has an opposite stereochemistry, which is S, and the opposite binding mode. You can see to oxygen or the catechol group, with reference to the active site glutamate 219, how they are oriented. And of course, the inhibitor was claimed to have a novel binding mode because stereochemistry and binding mode both are opposite. So what we did was we took that S-noradrenochrome structure, and that was one of the structures which was unstable in our simulation. And then we went back and said, look at the electron density. First thing when we read the paper and the structure, that they had used a racemic mixture of the ligand, and they had not explained why they chose a stereoisomer in the binding pocket. So what we did, we took both S and the R, and the shape of the density was such that there are 4 different ways each stereoisomer could be placed in the electron density. So we had this 8 possibilities for 2 stereoisomers of noradrenochrome in the binding pocket. So first thing we did was, all right, let's run the molecular dynamics simulation and see how many are stable. So what we found was that out of this 8, 4 were unstable, including the one which was in the crystal structure. Okay, and then we are now left with other 4. Now out of other 4, which one is the most stable? And again, then we go back to our calculation of relative binding free energy and see which is the lowest free energy state. So we build again a few closed thermodynamic cycles and calculate the free energy. And what we found that when we did all these calculations, the structure what we call DR, which is on the bottom right in the figure here, is the most stable in terms of thermodynamic free energy. Then we say, all right, that's good. So let's go back and compare now. Now this is what was originally reported, that the inhibitor has an opposite stereochemistry and opposite binding mode. But when we did the MD simulations and free energy calculations, what we find actually, the inhibitor actually has the same stereochemistry and the same binding mode. So again, this again just demonstrates another example where what we the binding mode we see from the X-ray crystal structure at a given resolution is not reflecting what it would be. And again, many of this data, of course, rely on the resolution you get in the X-ray structure. And in addition, if you have additional data of those protein ligand complexes coming from NMR and some other methods, they are quite useful and they can help you to deconvolute what is the stable binding mode in the structures. So this was just one of two examples where we found that the ligand was unstable, and then we could use computational methods and protocols and then work out, which is the most stable binding mode. So continuing with the PNMT system, another study they had done, actually, they had screened about 96 cocktails of the fragments using high throughput crystallography. Now what they had done, each cocktail contained 4 fragments of small molecules, and they found about 15 hits on those screening. And they identified 12 fragments based on electron density. So what they had done, actually, they took the cocktail of fragments, the hits when they found, what they did not go and further deconvolute that out of 4 molecules in that cocktail, which one is in the protein. They just look at the shape of the density and made a guess. This is what it should be. And then additional confirmation was done using ITC on some of the fragments. And they also did additional ESI mass spec. And they published overall 12 crystal structures from this study. When the student did the simulation of 12 structures, 11 were stable. So 11 did match what they found from the electron density, but one was unstable, and that was of interest, that what is going on with that structure. So we looked at the composition of that cocktail. It contained these 4 compounds shown here, F1, F2, F3, and F4. And there was an additional data that they could not detect some of these compounds by ITC, and this cocktail was not deconvoluted. So this is how the electron density looked, and they could place the fragment number 3 in the binding pocket and submit it to the protein data bank as 3kpy. Now whatever we did with this PDB 3kpy with protein complex and run molecular dynamics simulation, within few tens or hundreds of picoseconds, the ligand would just leave the binding pocket. We try to place the inhibitor in a different orientation, run multiple simulations starting with different conditions, but in all cases, it was just not stable. Then we thought, all right, okay, let's keep it aside. Let's look at the other fragments of this cocktail. So because the electron density was pretty large here, so we said, all right, F1 and F2, they look very small, forget. Let's look at the F4 and try to place it. But then when we try to put that 4th fragment in the binding pocket, we notice large number of clashes in the binding pocket because that ligand or the inhibitor was quite large compared to the volume of the pocket. And we said, all right, now that's not going to fit in. Then we took the fragment number 1, which was the next biggest. And that one was fine, stable in the binding pocket. But again, when we put that in the binding pocket, it still explains only the part of the electron density, not the whole electron density. Then we said there's something else in the pocket. Then we took fragment 2 by itself and run the simulation. And again, it's stable in the binding pocket. And we say, okay, that's fine, but it still does not explain the electron density. Then what the student realized that when we ran simulation with F1 and F2, they were binding to the different regions of the pocket, not exactly at the same location. So what we did was that, all right, then we placed both the fragments F1 and F2 together as we found individually and run the simulation. And to our surprise, that complex was stable. And then we again did some free energy calculations, and what we realized that actually the free energy of binding we get from individual fragments, and when we do it for the combined fragment is much higher or better. So we observe something called what we call cooperative binding. And based on that structure, what we did was we went back and re-refine the structure because we had access to the structure factors from the data bank. And then we said, actually, the electron density represents these two fragments in the binding pocket. And we submitted this coordinates as a new protein data bank entry to the PDB. So this is an interesting example where what happens, the raw data, what we call the structure factors from the X-ray crystallography is identical. But there are two explanations. One submitted by the crystallographers, which is PDB3KPY, and other one re-refined done by us with the PDB4DM3. And now here you can see the contrast, that how critical it is to look at the structures carefully. Now, one major aspect in the fragment-based drug design that you want to identify small fragments which can occupy different parts of your pocket, and then you link them to come up with the lead molecule. That information, if you look at now the structure on the right hand side, it was there sitting in that structure, but for whatever reason it was overlooked or not or misinterpreted, and here they actually lost in a way a potential lead they could have got by linking them. Later we wanted to pursue this further to test this experimentally, but that they group moved on to other projects from the PNMT and we could never do that. But again, this one just to demonstrate that same X-ray crystallography data can have two different interpretations which are widely different. So here I just summarize some of the studies we have done over a time and just showcase that what kind of systems we can't make a distinction based on the electron density, and we need to use molecular dynamics and the free energy. It could be the conformation of the molecule, it could be the tautomeric or the protomeric state of the molecule, it could be the structure or the relative orientation or the stereochemistry of the compound. So now what I will do, I'll now pick up on the what we call is the tautomeric or the protomeric state of the molecule as an example. To choose that one, what I did was, of course, if you look at the tautomeric definition, they are molecular chameleons. And those structures, of course, they can exist in a multiple state depending on the movement of the hydrogen atoms defined by certain rules. And the major question in the tautomers is that, for the drug design point of view, whether the tautomer which is free in solution and the tautomer which is bound to the protein, are they the same tautomer or different tautomer? And that can have a great implications for drug design in many cases. And one of the classic examples of tautomers in biochemistry is the sugars, whether they can exist in the ring structure or the open chain, and the ring also whether the 5-membered ring or the 6-membered ring, and have a lot of implications in terms of biology. So the example I have worked on and picked up for tautomerization is one of the extreme examples in literature, and that is called warfarin. That was an interesting compound. Now, warfarin was approved for treatment in 1953. Now, that was the time they observed anticoagulation activity in animals and immediately was approved for the human use, which you can't do anymore currently. This molecule is prescribed as a racemic mixture of R and S, and vitamin K epoxide reductase is the drug target, and it binds extensively to the human serum albumin and cytochrome P450. Now, if someone has been on warfarin therapy would know that if you are taking warfarin, your blood clotting time has to be monitored on daily or weekly basis, and the dose has to be adjusted because warfarin forms a lot of interactions with the food and food components and interacts with so many proteins here. So what happens, the warfarin level can fluctuate depending on your eating habit or the dose, and the dosage has to be adjusted. Because if it's overdose, it can lead to bleeding, and if it's underdose, it can lead to blood clot, and both the things are not safe. Now, what are the reasons for this complex pharmacokinetics and continuous patient monitoring? Now, there are new anticoagulants have come up in the last few years, but still worldwide warfarin is still the drug of preference, under certain conditions. So it's still being used. That Most of the warfarin structure, if you check in the literature or any database, they will show this chemical structure which is shown on this slide. So it can exist in neutral form and a low pH or the solid state or in abrasive solvents. And it can exist in anion or the salt and the physiological pH. Now the interesting aspect here is that each stereoisomer in the neutral form can have 20 tautomeric forms. So technically, when you take a warfarin tablet, at least 42 chemical entities are floating in your body and creating havoc. And we don't know which of them is useful, giving you the desired action, and which of them is having off-target interaction and giving you all possible side effects. And the big challenge here is that it's not easy to isolate all the tautomers by experiment. Now there are ways to trap and isolate. There's a limited data available, but not much. And most of the data from X-ray and NMR is pretty much convoluted. It could be more than one tautomeric state, which you can't go back and deconvolute or dissect. So this slide shows just 20 tautomers of R warfarin. And similarly, you will have 20 tautomers for the S warfarin. And here, the major tautomers are the 4-hydroxyenol and 2-hydroxyenol and the diketo. And the variation in the side chain where there's a keto group can lead to enol. And one of the interesting cases in the warfarin are the 4 structures which are called hemiketals shown at the bottom of the slide, that it can form a cyclize, like with the intramolecular reaction and form another ring. And when it forms another ring, actually introduce another chiral center here. So we got, diastereomers formation here. Now the big question is for us to understand out of this which are relevant, which are thermodynamically more stable or relevant when they are in free in solution and when they are bound to the protein. Now until unless we understand that, there is a less chance of having any success of designing or getting a better anticoagulant. So what we did was, because we are looking at the tautomers, we thought we will look at the whole process of that molecule formation in the water. So going from the left, what we are looking at the atoms which are at infinity. You look at the heat of formation, that what is the enthalpy required to form those, combine this atom to form this molecule in the gas phase or vacuum. And then you add entropy component, and then what is the free energy required to transfer this molecule from vacuum or the gas phase to the solution. So we are looking at the enthalpy of formation, entropy, and the free energy of solvation. And what we are trying to do here is that we are using these 3 what they call methods or the numbers and see if we are able to make any distinction between the tautomers. So in addition, what we did was for each step of calculation, we tried to test different methods and see which methods are able to reproduce some of the experimental data we know about warfarin tautomers. For heat of formation, we use 2 methods using direct method and the homodesmic reaction. For the free enthalpy of formation in vacuum, also we took into account vibrational and conformational entropy. And the free energy of solvation, we use fast implicit solvent method using ECM, Cosmo, and SMD. And then we also use explicit solvent and did the full free energy calculation. And the whole idea of this was to see which of these methods are able to give us distinction or difference between the tautomers. And here just showing an example of enthalpy of formation using an isodesmic reaction, which is like an artificial reaction where you know the experimental heat of formation of the components, and based on that you are able to recalculate or extrapolate for the molecule of interest here. So first thing we did was look at the heat of formation using 2 methods, direct and homodesmic reaction. Now again, we did not have much of the experimental data here, but the thing we noticed that with these 2 methods, direct and homodesmic method, what we are getting is an opposite answer. Direct method is telling that the tautomer number 1 is the most probable or the most stable, and tautomer number 17 is the next likely tautomer in the population. But if we do this calculation using homodesmic reaction, we get the opposite trend. where the cyclic tautomers are 17, 18 are the major, and the open chain, which is the first one, is the minor. So what we next we did was, and just show the structure here for those two, 1 and 17. When we look at the entropy contribution from the vibrational and the conformational entropy, and say, let's see, do the correction and look at not only the heat of formation, but the free enthalpy of formation. So when we did the free enthalpy of formation, of course, the numbers are all over, but we at least get the similar trend where in the gas phase, it shows that the open chain tautomer is the most stable or the most likely. And the next most likely, at least for the homodesmic reaction, is the cyclic tautomer, which is 17. Then we say, all right, this is fine. Let's look at the next component, because we looked at only the heat of formation and the free enthalpy of formation. What happens in solution? So we looked at the PC and solvent model and calculated the solvation free energy, and we got a trend where the tautomer number 6 was the most stable across the methods. Now we knew from the experiment that when the NMR is done DMS for this compound, only the cyclic compounds, the 17 and 18 are observed. No other tautomers are observed. We knew log P in oxygen and water. We knew solubility in water, heptane, because it's highly soluble in DMSO and sparingly soluble in water and heptane. So based on this data, we know that the free energy difference between DMS and water is roughly kind of 3 orders of magnitude. But we don't see that trend here. And then we know the free energy difference between hexane and water should be kind of similar range. But again, with the PCM, we are not seeing that kind of pattern. The same thing applies with the Cosmo and SMD. So, and then again, we did the explicit free energy calculation, and again, we did not see any trend. So next, what we did, we combined the whole process, not in isolation, but we look at the enthalpy of formation, take into account the free enthalpy, and then take into account the free enthalpy of solvation and what we call the free energy of solution. Then what we find for the 3 different solvents, we get this relative trend. And now that trend kind of matches with what we know from experiments, from small molecule crystal structure. We know these are the 2 tautomers we can crystallize. These are the tautomers which are observed in NMR. And this data kind of mostly represents or reproduces what we know by experiment. Then we say, all right, to distinguish these tautomers, we need to take into account the whole process, the quantum mechanics, the heat of formation, as well as free energy of solvation together, and that can give us close to the correct answer. So we knew here that there are certain tautomers, for example, have a relative difference in a particular range. So again, we look at this 20 tautomers of warfarin in water, and we notice that the 2 tautomers, which is 1 and 2, and 17 and 18 here. Now these tautomers are the ones which are within the 15 kilojoules per mole for energy difference in solution. Now when we look at the warfarin crystal structures with vitamin K epoxide reductase or albumin or cytochrome P450, the binding affinity is in the range of micromolar, which is in the roughly 30 kilojoules per mole. So based on that number, we can now guess here, and we know the pK is 5 for warfarin. So these are the 4 tautomers which are likely to be present in the binding pocket, considering the relative energy difference. And now again, R and S enantiomers won't be different in water because water is a uniform medium, but that can make a huge difference if they're in the protein binding cavity. So based on this study, we know that out of 40 or 42, actually there are 8 tautomers, 4 for R and 4 for S warfarin, more relevant or possible in the protein binding cavity. So at least from that, we are able to narrow down to the out of 42, at least we know 8 are possible in the protein binding cavity. Now the interesting thing is, historically, most of the drug design efforts for warfarin or the anticoagulant have been focused on the tautomer, which is the first and the second, which are open chain tautomer. not much attention has been paid on the cyclic tautomers, which are 17 and 18. So again, this also opens up a possibility that maybe these are the tautomers which could be exploited and design point of view, where we should be able to lock those cyclic tautomers, and we should be able to get warfarin derivative with the higher potency and the lower side effect, which may require less monitoring. And again, the whole purpose of this study again was to demonstrate that how individual tautomeric state of a molecule could be critical for the drug design perspective. To summarize what I presented today, that we show that we can use computational approaches like say molecular dynamics simulation and quantum mechanics calculations and free energy in combination with appropriate parameters so that we can understand what is the exact state of the molecule, what is the conformation, what is its orientation, what is the stereochemistry, which protomer, which tautomer, is there only one binding mode or more than one binding mode which are thermodynamically feasible and which could be identified using this technique. And again, this once we are able to identify this will be very crucial for designing the next potent molecule. With that, I would like to acknowledge a lot of people here. So most of this work I presented today were done under mentorship of Professor Alan Mark at the University of Queensland. And here are the students I supervised over last few years and who have directly and indirectly contributed to the work I have done. And there are some of the group members in my previous group. Currently, I work at the Institute for Glycomics, Griffith University, under mentorship of Professor Mark Wallenstein, and he's one of the co-inventor of Relinza, the drug for the flu. Got a lot of funding from Australian Research Council and competing resources from National Computational Infrastructure Australia and QSAFE in Queensland and Griffith University. A lot of work presented is based on the development we have done on the ATB, Automatic Topology Builder. And I would like to special mention for two people here, or two professors. Professor Ivan Skutino, who from my BPharm days, had a lot of discussion and interest in chemistry, eventually led to exploring the area of computational and NMR structures. And a lot of quantum chemistry I learned from Professor Bhartam, and all have been kind of a foundation for what work I have done in last 10 years in Australia. And thank you very much, all of you, for your listening and attending, and standing by after such a long day. Thank you.
Thank you, Alpesh. There is a question. The question is that we have PDB structures available between 2 to 3 angstroms.
Right.
So, how much importance we should give to the resolution?
Okay. So
initially, what I understand from this question
yes,
is that most are within the short range.
Right.
So how do we shortlist?
So what happens is most of the structures, of course, are medium resolution, and one has to remember the structure which are very high resolution. When we say 1 angstrom or 0.8 angstrom, that does not mean the entire structure has that resolution. That is the only best resolution in the some part of the structure, but the ligand may have 2 angstrom resolution. So having 0.8 angstrom resolution does not guarantee your ligand will be correct. And again, people have to remember that when you take a structure from protein data bank, it's just one representation or explanation of that data. People need to run some validation on those structures and check before doing anything with those structures. Sometimes as simple as when you look at your chemical intuition or ability, and it will make sense that, this does not look right because how two negatively charged groups are pointing against each other. This is not practically possible. But extra refinement program may not take that into account. And again, what happens? Is my screen still on, Hemant? Yes or no? No. I can't hear, sorry.
Yes.
Yes. So that was my last slide, which I just wanted to convey that the software and program, of course, are meant for a purpose, but at the end of the day, it's your ability and intuition as a scientist, which counts, and you go back and check. If you know that if something looks inappropriate or does not make sense, of course, then you should go and double check. So I'm not saying that all the structures are incorrect, but don't put blind trust on the structures from data bank.
Yeah, this was an example. So thank you, Alpesh. Thank you for joining us. And all the participants, so today's session we will close. Tomorrow we will have separate link has been provided to you. If you see our email, for today's session there was one link, and for the tomorrow session we have a separate link. So do join us tomorrow, and tomorrow we have another interesting set of people giving their insights on the computer-aided drug design. Thank you very much. See you tomorrow.
Thank you.